Hello, I am going to give a mini lecture on 16.2, and it turns out that this will be like the third time that I've recorded this, because when they're too long, I can't get them uploaded to YouTube. YouTube can't process them or something, so they have to be less than 8 minutes long. So I've got my timer, and it's going to make a weird woo 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 when 8 minutes is up, and I'm just going to have to stop talking and stop the recording at that point. So this is on the beginning of 16.2. I've already done the second part and already posted it to Moodle. So I'm going to have to try to remember where this one ends, where the first part ends. So let me get started. So first of all, I need to apologize that the rendering of the PowerPoint on my iPad is kind of goofed up. For one thing, superscripts don't show up as superscripts, so the NAD plus is looking wrong. And also, what's the deal with the blue title being overlapped with the black words? But I guess we can see anyway. So 16.2 says NAD plus is regenerated from the metabolism of pyruvate. Okay, so this is about glycolysis. Glycolysis starts with glucose and ends with pyruvate, but it can't continue by itself. Pyruvate has to be converted to something else that will allow for the regeneration of NAD+. NAD+ is an important coenzyme that is part of reaction 6 of glycolysis. So as an introduction, a quote from Stryer says, "An energy converting pathway that stops at pyruvate will not proceed for very long because redox balance has not been maintained." So NAD+ and NADH are always part of a dehydrogenase, and they're always redox reactions. The dehydrogenase always catalyzes redox reactions. And reaction 6 is the only redox reaction in glycolysis, and it uses NAD+. And the cell has only a limited amount of NAD+, so glycolysis cannot continue for very long without the NAD+ being regenerated elsewhere in the cell or through another pathway. So glycolysis can continue if pyruvate is changed into either acetyl-CoA, which enters the citric acid cycle, and the NADHs made at the citric acid cycle go to the electron transport chain, where there's more redox reactions that convert the NADH into NAD+ at the electron transport chain. So that's one way to regenerate NAD+, but that can only happen when there is ample oxygen, O2, in the cell that this is happening in. If our muscles, for example, if we're working really fast, like we're sprinting, we're breathing hard, and our heart is pumping the oxygen through our blood vessels to try to oxygenate the cells. But if our muscles are working too fast and glycolysis is going too fast, oxygen, the heart can't keep the oxygen getting to the cell fast enough, and so the cell is actually going to be low on oxygen. But glycolysis can continue for a short amount of time without oxygen anaerobically, and that's because of fermentation reactions. So depending on the type of cell, there's different types of fermentation reactions. Yeast can do alcoholic fermentation. Our cells cannot do that type. Our cells do lactic acid fermentation. So let's go on to the next slide. Figure 16.4 is a figure of the different types of fermentations and of pyruvate, or the different fates of pyruvate. Two of these, the ones on the left and the center, are the fermentation reactions, and the one on the right is the reaction that happens when there is enough oxygen in the cell. So the one to the right, pyruvate is changing into acetyl-CoA, which further oxidation reactions will then regenerate the NAD+. The one on the far left is done by yeast and not by our cells, where pyruvate is decarboxylated to acetaldehyde, and then redox reaction by a dehydrogenase reduces the acetaldehyde to ethanol. At the same time, NADH is the reducing agent, and it gets oxidized to NAD+. So again, this is the way that yeast can continue glycolysis even in the absence of oxygen, because NAD+ is regenerated by this fermentation reaction. Now, this ethanol is the alcohol that is found in beer and wine, and so the brewing industries and winemaking industries are very interested in this fermentation reaction, and that's one reason it's in our book, because it is a very well understood fermentation reaction. We're going to look at pyruvate going to lactate because that's what happens in our cells. And part 2 of this has that spelled out very carefully, and why we need the NAD+. Going on, here again, when there's not enough O2 present in the cell, pyruvate can be reduced to ethanol, or pyruvate can be reduced to lactate. And notice in both of these reactions, NAD+ is one of the products. So lactate fermentation is necessary to generate NAD+ under anaerobic conditions. So when you're sprinting or your muscles don't have enough oxygen, they can continue to do anaerobic glycolysis. And actually the lactate, you can think of it as lactic acid. It's lactic acid fermentation. And the acid produced actually is in the cells and therefore lowers the pH of the cells. And so the buildup of that pH or that acid in the cells could damage your cells. So glycolysis has to stop at some point, and that's when you feel real fatigue in your muscles and you just can't go any further and you have to stop. That's because once the pH gets low enough, phosphofructokinase, the main regulated enzyme in glycolysis, is inhibited and slows down due to the low pH. And I'm going to stop this recording now, and you can pick it up again in part 2 of this section 16.2.